Perfect, thanks. All right, everyone, thanks for coming here and taking a second to hear what I got to say. So I'm going to be talking to you about the predictive biomarkers in the MAP kinase-driven pediatric gliomas. And so just for the sake of the recording, I'm Dan Muldoon. I'm a research technician at NYU Langone in the Nicolaides lab. All right, so just to give you some background on the pediatric gliomas. So neuroepithelial tumors are found about 4 in 100,000 children. Polycystic astrocytomas are the lower grade gliomas, and they're found about 1 in per 100,000. And then the glioblastoma is about 0.17 per 100,000. And treatments that you've got surgery, which is gross total, where you take everything out, and subtotal, where you just take part of it out. Usually, especially for glioblastomas, you'll get chemotherapy like temozolomide, get radiation, and then more recently we've got targeted therapies such as vemurafenib, which is a BRAF V600E inhibitor. You'll hear a lot more about that coming up. So therapeutic progress, we've got dabrafenib, which is another type of BRAF V600E inhibitor, and also selumetinib, which is a MEK inhibitor. So looking at the survival probabilities of the lower grade gliomas, we can see gross total resection, we can see survival probability around 90% after 250 months. And subtotal is over 200 months. After 200 months, we still get around less than 40% survival probability. So there's quite a bit of need still for when tumors are subtotally resected, and there's a lot of reasons why that may be. It could be that it's in a very difficult spot to reach. It could be that the tumor is very invasive and that it's spreading diffusely. There's a lot of different reasons why it would be subtotally resected, and so these therapeutics are very important to kind of bridging this gap and get this survival probability up for these kids. So the genetics of the low-grade gliomas, I'm going to be focusing on BRAF V600E, and these are all different areas of the brain, and we can see that BRAF V600E is pretty prevalent in the hemispheric portion of the brain, and there's other areas too, midline, where BRAF V600E mutations are very important. So in high-grade gliomas, as you can imagine, it's much worse. So overall survival after 5 years is less than 25%. And event-free survival is around 15% after 5 years. And that's with treatment, that's with hitting it with everything that you can. The genetics of this is much more diverse, and BRAF V600E is around 7 to 12% occurs in these high-grade gliomas in kids. This is just the frequency of BRAF V600E in the different grades. So there's 4 different grades, and this is at least kind of a more global look at where BRAF V600E is occurring in these tumors. So why is BRAF V600E important? You've heard me talk a lot about it, and it involves the MAP kinase pathway. So the way that works is you've got the receptor tyrosine kinase here on the cell surface, and that transduces through this pathway involving RAS, and then signals to RAF proteins, specifically BRAF here, and then onwards to MEK and ERK, and then to transcription factors that modulate cell growth, so survival and motility. And BRAF V600E in low grade is about 5 to 10% of all of those gliomas. And then, as you heard earlier, 7 to 14% of the high-grade gliomas have a BRAF V600E mutation. And this BRAF V600E mutation is completely independent of any RAS signaling. So it functions as a monomer, and it acts independent of the pathway, of RAS, I should say. So what we're looking at in our lab is what is going on with resistance. So there's 2 different types that we can talk about. There's intrinsic, where the tumor is already resistant to the treatment before you even use it. Another is acquired, where you hit the tumor with the drug, and then over time, it may work initially, but over time it ends up not being as effective. So we're focusing right now on the intrinsic portion of this, and looking to see what mechanisms could bring about intrinsic resistance. So we have a limited understanding of what's going on with these resistance mechanisms. Just in 2018, we've got it was found that the BRAF L514V mutation was one of the resistance mechanisms that came up after. So this would be an acquired resistance, but the initial resistance is still relatively unknown, intrinsic resistance. And just to showcase the kind of scope of variability here. So this is a waterfall plot from MRI scans showing that we've got the red here indicating that these are BRAF V600E mutations and within the tumor. And after treatment with this MEK inhibitor, this BRAF V600E tumor increased in volume 100%, while this one all the way on the other end decreased almost completely. So there's quite a lot of variation, and so looking into these drivers is very important. So the goal of our project was to elucidate biomarkers that correlated with efficacy with BRAF inhibitors and MEK inhibitors in these BRAF V600E glioma cells. So to jump into the project here, so these are all our kind of tools, our cell lines that we used. 8375, of course, is not a tumor or a glioma, but it is BRAF V600E, and a lot of these studies are built upon the melanoma cell lines, and a lot of what we know about how to work with BRAF V600E in gliomas is from what was found in melanoma treatments. So we've got all these other glioblastomas or gliomas of all different kinds. Some are adult, some are pediatric, but all have the BRAF V600E mutation in some form or fashion. Another big one is P10 we see a couple times here. And so this is kind of our foundation here. So with that, we wanted to approach things a couple different ways. We had a supervised approach and then an unsupervised approach. Supervised, looking at the receptor tyrosine kinases through immunoblot, MAP kinase pathway, and cell cycle proteins through immunoblot. And then unsupervised, which I'm sure we're all much more interested in, is the RNA-seq. And maybe we'll talk about the ingenuity pathway, but then also the phosphoproteomics that we'll eventually be working with at NYU at their proteomics core. So diving into the work that we did, so specifically Carly, who's in here, so thanks again for all this. This was almost entirely her work. So what you're looking at here is a plot that each dot indicates a single IC50 from a cell viability study, where you take a 96 well plate, you put a bunch of cells on the plate, and you see where the 50% mark of how many cells are still viable, 50% of the cells viable after treatment with this drug, PLX4720, at what dose are 50% still viable. So she did this what feels like hundreds of times, but we've got a nice curve here indicating the log nanomolar IC50 of PLX4720, the BRAF V600E inhibitor, and so we can kind of see that there's an increase in resistance to this drug, and we use this as going forward the base of the rest of our analyses. So this too, with a MEK inhibitor, GDC0973, we've got the same kind of situation with the IC50s and the cell viability assays, and we've got the kind of curve here of least resistant to most resistant cell lines to this drug. So what we found is there's evidence of the PI3 kinase mTOR pathway in regarding innate resistance to these drugs. And the way we found that, so we looked through the immunoblot, and I'll kind of run through this. So you know, we're not too interested in this, but we ended up finding phospho-AKT increase over as you increase or as you decrease sensitivity to the MEK inhibitor. So, or you could say as it increases resistance. So as you increase resistance to this drug, you can see the phospho-AKT increases, and we can look at this graph here. So we've got RAS that if this was not a mutated BRAF, there would be a line here indicating RAS also transducing through BRAF, normal BRAF, but also it also transduces through PI3 kinase and then onwards AKT mTOR pathway. And so implicating AKT shows implicates the RAS transducing through PI3 kinase mTOR pathway and causing resistance potentially. So we looked at more proteins related to the PI3 kinase mTOR pathway, such as phospho-PRAS40. And this is just an abridged blot where we took the two the two least resistant versus the two most resistant. And we can see that we see again the phospho-AKT increases, and then the phospho-PRAS40 increases, and then other proteins in activity of proteins in the MAP kinase pathway did not indicate that they increased or decreased with increased resistance. All right, so on to what we're most interested in. So this is the RNA-seq setup. So we've got the 8 cell lines, and we took each cell line in triplicate, so biological replicates of the experiment. So they're not treated with anything. They're just cell lines, you know, right out of the box, more or less. And so we wanted to compare the least resistant to the quote most resistant, and do the differential expression analysis on that. So this is just the kind of synopsis of how we got through that. We got the 50 base pair end reads through the NovaSeq, and we got about 35 million reads per sample, processed it through the high performance computing servers here at NYU. And then they were trimmed through the Trim-O-Matic, lined with RNA star, and then we got the gene sample counts through feature counts, and then we used those feature count files to feed through DSeq2 and see what our differential expression was. So here's the design. I'm just going to showcase the BRAF inhibitor resistance analysis for now. So we looked at the least resistance. So we considered those to be BT40, ATRT, and MCG1, and the AM38 cell lines, and then most resistant being TBTRG, B76, NAF, and GPM110. So here's our PCA. So we could see that the most resistant to BRAF inhibitor cell lines are clustered here in this area, and then we've got a couple of cell lines that are the least resistant cornered here, AM38 up here, and then interestingly, this NMCG1 is clustering closer to the more resistant cell lines, and we'll discuss why that may be the case going forward. So this is kind of one of the quality control plots, the concentration of counts over the sum of the counts. This is part of what I'm kind of struggling with to see how viable this analysis is. So this, from what I know, what I can tell is that this is pretty normal, but we've got this up here that I can't really make heads or tails of why this would be the case. So this is the max count over the sum, and then the sum of the counts per gene, and we've got a bunch clustered here. So we can discuss that later on, what's going on there. All right, so here is our volcano plot. So it's least resistant to most resistant to the BRAF inhibitor, and we can see that we've got GPC4, which is found to be increased in the most resistant cell lines. So we've also got DUS15. So we were, which I didn't show, we were looking for a bunch of different DUSPs beforehand, the kind of supervised approach, and we checked several. None of them seemed to be correlating at all. So to see a DUSP pop out kind of popped our curiosity. So we looked more into that one. Here is a the heat map of the so this is going from the most downregulated to the the most least expressed to the most expressed comparatively, and out of the 1,000 most significant genes, yeah. So then we will look through different gene set enrichments analyses. One of them being this one here, the Hallmark study, and nothing really stuck out to us necessarily. I'm trying to remember what it was, if there was anything. Yeah, not too much. Of course, here's the KEG, the neuroactive ligand receptor interaction, which is interesting considering we've got some colleagues looking at how electrical impulses in glial cells are implicated in gliomagenesis. So that's interesting. All right, and then gene ontology. So this was very successful, I would say. So we found that the regulation of PI3 kinase signaling was one of the most enriched sets of genes in this analysis. These are the top 10 here of the biological processes. Here's the one that we saw previously. This was just the whole hierarchy kind of spread out. And now we've got the top 10 here. So we've got the PI3 kinase, and then we've got some, of course, neuron and glial related ontology sets, synapse organization, axonogenesis, etc. And then we did a quartile study. So I'll just speed through this for the sake of time, but we looked at the BRAF inhibitor, looking at the 2 most resistant versus the 2 least resistant. So we got the MAF and the GBM most resistant clustered here, and then the 2 least resistant on the left. And we did another volcano plot, and we cross-referenced the genes, and 2 of the big ones that came out were algal-S3 and PAPA, whatever you want to call it. So we looked at those genes. And yeah, here's another KEG, and the Hallmark gene set study, nothing interesting here. Here's another heat map, looking at first quartile versus last quartile. Didn't find anything significant that interested us. We've got our top 10 here. Interestingly, not too much related to the brain. Yeah, so here's our blot. We used the unsupervised approach to find algal-S3. Looks like it increased over time, or as you increase BRAF inhibitor resistance. DUS15 also increases with BRAF inhibitor resistance. And yeah, so I'm going to kind of wrap things up here. I just had some, kind of broad questions for everybody, if, depending on where everybody's at. But so we're thinking of how to kind of parse this to better analyze it. And one way was maybe doing some kind of linear regression analysis for differential expression. I don't know how viable that is. I don't know if I'm trying to figure out if people have done it before. And for the most part, haven't really been able to pick anything up. Like, it seems like you can do it, but there's not a whole lot to say that you can find anything valid about your analysis, like the downstream kind of like your log twofold change and all that. But we can discuss that towards the end here. So evidence of PI3 kinase mTOR activation leading to resistance, and then algal-S3, GPC4, and DUS15 could be potential biomarkers. So I just wanted to thank the lab for all the work that we've been doing. This big team effort. So especially Carly, thanks again for all the work that she's been doing. So let me know if you guys have any questions. And I also have my own questions. So let's make this a dialogue and try to help each other out.
Okay, excellent presentation. Thank you so much, Dan. Does anybody want to go ahead and jump in with any questions or comments? I have a lot, so I want to let other people. I want to let other people have a chance first before I jump in. Okay, well, I can get us started then. So, I thought this was a really interesting presentation and really excellent data. I really like how you presented everything. I think the thing that struck me most was actually those slides where you said, "Oh, didn't find anything interesting." Actually, I thought those were the most interesting slides of all of them because of the things that we study in our lab. Because a lot of the things that we would have anticipated you did find actually. So I'm very interested in hearing about some of your thoughts on, for example, how you saw like the oxidative stress response and EMT and that sort of thing. If that's something that you guys are pursuing at all.
Yeah, so as far as that stuff goes, not in our lab particularly. That's I think kind of the division of labor is kind of fixated on the different, just particularly the MAP kinase pathway and the PI3 kinase mTOR pathway, and we've kind of just been pursuing that as of now, as far as resources go. But yeah, it's absolutely not unthinkable to see EMT and oxidative phosphorylation seeing increased activity or activation of those genes. And that would be definitely something worth pursuing. Let's see, what were the ones that popped out?
Yeah, and they came up. Oh, sorry, go ahead.
No, I was just going to say they came up pretty regularly, either whether or not we did the quartile differential expression or if we did the full 4v4 differential expression. So yeah, that's a good point. That's a really good point. That might be something to look more into.
Right. I was just curious about that. So the overall the question I think I have most is how does this relate to the level of the genotype, right? So how does this relate to the mutations that you see in the clinic? And can this be used as a way to determine how best to treat those patients?
Yeah, that's a good question. So so the kind of next step that we plan on doing is doing staining on patient samples of IHC slides of their tumors and seeing if we can pull any of the proteins that were significant out of this study and see if we can get them in those clinical samples. And going forward after that, it's kind of like, okay, so now we know we have these potential biomarkers. What are the biological mechanisms that make them biomarkers, and are they related to something that we haven't seen, we don't necessarily know about? But clinically, what this means is it further characterizes these tumors, these gliomas, and that's important for prognosis. It's important for if you go in, you get your tumor sampled, and you get your biopsy, and it's one of the biomarkers that show resistance comes up, and you're like, oh man, well, as your doctor, you're saying, okay, you don't necessarily want to start them on a drug that is not going to work potentially, so, or be effective. And so it would fast track that person to a different clinical trial and skip the first phase. Why would you put somebody on a BRAF V600E inhibitor if you know they have a biomarker that tells them that it's not going to be effective, And so it gives them, months, potentially years of, not going through chemotherapy and just going on to a, they'll have to go through chemotherapy, but down the line, it would save them time and give them a chance to have access to something, it's more knowledge. So they can just skip the BRAF-V600E trial, go to a different trial of a different drug that may be more effective for them.
Absolutely. Absolutely. Very cool.
Yeah.
Okay. Did anyone else want to ask a question?
I wanted to hear what you guys were thinking of. I don't know if you guys can still see my slides. I can't get Zoom back up.
Yeah, we can see it's in like the
Oh, okay.
yeah.
Good.
We can't see the full screen though.
Okay, let me see.
Oh, there
if
you
I
go.
can drag you back to my questions. So my idea, kind of, which I brought up earlier, the linear regression analysis on this. I don't know if it's feasible. I don't know if, like, I'm scratching my head how to get it done. Because you can run it through DSeq2, but like I was saying, you can't really. How do you interpret the downstream results? I don't know. So the linear regression would be, we take those IC50 numbers, so say from BRAF inhibitor, and so you got one that the IC50 is 10 nanomolar. Then you got you go factor by, or number by number, use that as one of the factors, the number, the IC50 number, and all the way to the MAF, which is 30,000 nanomolar on the BRAF inhibitor, and use that as a way to differentially express the genes. So I don't know. I don't know if that even makes sense, or if that's doable. I don't know if any of you have experience or thoughts on that. I'll take anything.
So have you looked into using the log ratio test?
I've heard of it. I know you can do that in DSeq2, right? That's one of the options.
Right. So you can run DESeq2 as either a paired comparison or as a log ratio test, which is kind of like the DESeq2 version of an ANOVA, like something that's looking at some multi-level factor like IC50 or time or even groups, and seeing if there is a significant change across that factor. And so you can use the log ratio test for like time course, and anything you can use for time course, you can pretty much always use that also for things like different IC50s, something like that. Mm-hmm. The other thing which you could do if you had more samples, I'm not sure if you've got enough. Well, you might have enough samples to do this. Is to do just correlation. So taking all of your data, doing like some variance stabilizing transform, and then seeing what the correlation of each gene's expression is with that, with those IC50 values. But probably the log ratio test is a bit more what DESeq has in mind, and therefore probably a little bit more mathematically correct.
Gotcha. All right, cool. I'll give that a shot then.
Yeah. And actually, you brought up another point, which was the in the beginning, you were saying that you were seeing the max count value, some artifact. Yes.
Yep. Let's see here. I got it somewhere. Oh, there we go. Yeah. So we got going on up here. Not sure what to do with this.
So this is the sum of the counts per gene, and then the max count. So are these log transformed?
Yeah, I believe so. Yeah.
That's why. Yeah, that's why you're seeing that.
Okay.
Yeah, because you probably did a log n plus 1 or log x plus 1. So wait, would that be right? So yeah, it does make sense that you have like a pile up at that one value. That's basically 0. Yeah.
Gotcha. Yep. Yep. Yeah, you're right. Yeah. All right. Good to know. So I guess with as far as pulling those out of the analyses, if those would, or keeping those in, I think I don't think this necessarily changes any of the downstream results. So I think I'm just gonna keep it as such.
Yeah, from what I can see, your analysis is very good as is. I wouldn't think that you would need to change a whole lot. What I was curious about was your PCA, that one cell line that was more similar
Yeah.
to
Oh
the
yeah.
BRAF resistant ones. I'm curious
Yep.
if you guys have kind of come up with an idea of what that is. Also, what cell line is that? Is that
So
NMGC1?
NMCG1, yeah, that's just a, I think it's a glioma of some kind. I can't remember if it's adult or, let's see, it is adult. Yeah, so it's got an CDKN2A deletion. It's got NF2 frame shift, so it's got a pretty heavy load of very intense mutations. And I'm glad you brought that up because the analyses, let's see if we got it here. Yeah, so when we look at the MEK inhibitor, NMCG1 is one of the most resistant cell lines to the MEK inhibitor. So I think the fact that we see it hanging out closer to the resistant cell lines in this, unsupervised kind of approach, it's not, it's makes sense. Yeah, that it would be over there. Yeah, and it's kind of, that's why I think the linear model might be a little bit better because we're missing, like, if I just arbitrarily call 4 of them not resistant, 4 of them resistant, it kind of loses the nuance of, well, this is sort of resistant, it's not really resistant, but we don't, for the sake of the analyses, just to do the factor analysis, the 1 versus 0, we got to put it somewhere. So yeah, that's a good point. Yeah.
What I thought was just so interesting about this whole thing is it seems like the less resistant ones can kind of be all over the place. There's a lot of cellular states that they can sort of inhabit, it looks like. But then once you become more resistant, it's like you fall into one sort of cellular state. And
Yeah.
maybe it's characterized by some aberrant mTOR activation and perhaps some mesenchymal phenotypes of some sort.
Yeah.
I just thought
Yep.
this was just very, very interesting, actually.
Yeah, that's a good point. Yeah, like you're saying with seeing the keg pull out, or the hallmark studies show, or the gene set enrichment analysis show the EMT stuff. That might be something interesting to look into. Yeah, and I would also say this AM38 cell line that we've had is annoying to say the least. It's genetically, it, like, you can passage it so many times and then it just gets absolutely unusable, and it's hard to, we've had so many problems with the cell line, so seeing it kind of hanging out up here outside, away from the other 2 that are more sensitive to these drugs, it's not ideal. And unfortunately, these are the tools that we have. So it's kind of like, well, I'm not going to take it out. We did it going forward. Let's just see what happens. So.
Absolutely.
Sorry, guys. I wanted to follow up on the max count, the previous slide.
Let's see this guy here. Yeah.
Yes, that one. So I remember some time back, Henry, we talked about these the outliers here. Are they the ones usually referred to as the values which the very less 0.00 something values that R cannot be able to plot, or these are different ones?
So I think you're thinking of volcano plots where you have the p-value is so small that it just defaults to 0. This is different. So this is that he's looking at in the actual raw count data. Well, it's log transform count data. the sum of the counts for each gene by the maximum count in that gene divided by the sum. So I think that's why we're seeing it like this. But yeah, no, this is a different issue from that.
Oh yeah, yeah, all right, absolutely.
All right. Were there any other questions or comments? Okay, so Liz F asks, which variables would you use for a linear regression?
The IC50 value. So let me show you here. Pull it up. Man, I got a lot of slides. All right, so so IC50 value here, just where 50% of the cells are still viable after you treat the cell line. So like BT40 here, we've got a range going from at least the test was something like 0.01 nanomolar all the way up to 50 millimolar or something like that, and we were able to get a curve. And the curve, you do the analysis on it, and it tells you where in the middle of the curve there's only 50% of the cells left in your plate, and that's where your IC50 is. So we've got, for this example here, BT40's got 500 nanomolars worth of. If you hit it with 500 nanomolars of this BRAF inhibitor, there's 50% of the cells left. So as you go along here, you can see that this IC50 value increases. So we would use the IC50 value in the linear analysis. So BT40 we would say it would be 531, and then ATRT would be, 1700, and etc. So we can use that as the linear regressions variables. What do you ever call them? Covariates. I don't remember. It's been a while.
All right. Well, all right. So excellent presentation, Dan. I apologize because I actually have to jump to another meeting now, but let's all give Dan just a real quick virtual round of applause here. Yes, thank you.
Thanks, everyone.
And yeah, all right. So